I want to start today by taking you to Paris. We're going back to the mid-19th century, specifically to the Salpêtrière Hospital.
Oh, the Salpêtrière.
Yeah, it's this massive, crowded, slightly damp institution, but is arguably the birthplace of modern neurology. And walking the halls is a man named Jean-Martin Charcot.
The legend himself. They called him the Napoleon of the neuroses, just a giant in the field.
Exactly. And Charcot had this incredible eye for patterns. He could look at a patient's tremors or the way they walked and, categorize them. And his name is actually the common denominator in two very, very different diseases that we're doing a deep dive on today.
Right. And for over 100 years, neurologists have sworn these two are completely different animals.
Completely. On one hand, you have Charcot's first big discovery, amyotrophic lateral sclerosis, ALS.
Lou Gehrig's disease.
Lou Gehrig's disease, yeah. It's aggressive. It destroys the motor neurons in the central nervous system, so the brain and spinal cord. It leads to paralysis, respiratory failure, usually within, 2 to 5 years. It's a terrifying diagnosis.
Absolutely. It's a systemic collapse of the motor network. It moves fast, and the prognosis is it's heavy.
And then on the other hand, you have Charcot-Marie-Tooth disease, or CMT, which, by the way, has absolutely nothing to do with teeth.
No teeth involved. It's named after Charcot, Pierre-Marie, and Howard Henry Tooth.
Right. And CMT is a peripheral neuropathy. So it affects the nerves in your hands and feet. You get high arches, maybe hammer toes, or that characteristic foot drop where you have to lift your leg high just to walk.
And this is the key distinction, people live with it.
For decades. It's chronic, it's slow, and it's rarely fatal.
So you have one that is this rapid, lethal firestorm in the brain and spine, and another that's a slow burn in your toes and fingers. Historically, no doctor would put these in the same file folder. One is a crisis, the other is a condition.
But today, we are ripping up that file folder. We're unpacking a fascinating paper from 2026 by Abdellatif, Aynaj, and Petri Kursula that essentially says, we've been looking at this all wrong. These distant cousins are actually genetic siblings.
And they've brought receipts. They identified 13 specific genes that overlap between familial ALS and CMT.
13 genes. I mean, that is not a coincidence. That is a pattern. And that's our mission today. We are going to find out why your neurons essentially have a trash management problem, why the sheer length of your nerves is your biggest vulnerability, and why mother and son can have the exact same genetic typo but live two completely different lives.
It's a fascinating look at the machinery of neurodegeneration. We're going to, open up the hood of the cell and see exactly what parts are breaking down.
So help me bridge this gap. If I'm a clinician, I see two completely different diseases. One kills quickly, one disables slowly. But you're telling me the source code is overlapping. Where do we even start with that?
We have to zoom in, way in. We have to forget the symptoms for a second, forget the foot drop or the paralysis. We need to look at the maintenance crew inside the cell. The paper categorizes these 13 overlap genes into specific jobs: RNA metabolism, axonal transport, mitochondrial function.
And the big one, which I know we want to spend some time on, protein homeostasis.
Right, proteostasis, which is really just a fancy scientific way of saying keeping the house clean.
I like that. But let's get specific, because keeping the house clean sounds kind of passive, like sweeping dust. But inside a cell, it's a little more violent than that, isn't it?
Oh, much more violent. Think of your cells as high output factories. They are just churning out proteins constantly to do all the work of staying alive. But proteins are these fragile 3D structures. They can break, they can fold up the wrong way, they can get sticky.
Okay.
And if you don't clean them up immediately, they turn into toxic trash. They clump together and, they choke the cell.
And this is where our star witness comes in. The paper focuses really heavily on a gene called VCP.
VCP, velocin-containing protein. If you want to get technical, it's also known as P97, and it belongs to a family of proteins called AAA ATPases.
Okay, I see ATPase in there.
Yep.
That implies energy. This thing actually burns fuel.
It burns a lot of fuel, and this is crucial to understanding why it matters. VCP isn't just a trash bin, it's an engine. Structurally, it looks like a hexamer so. Imagine 6 units arranged in a ring. Almost like a donut.
A molecular donut.
A molecular donut that acts like a crowbar. Its job is to find those misfolded, sticky trash proteins, grab onto them, and physically pull them apart. It uses the energy from ATP to thread those proteins through the donut hole, unfolding them so they can be destroyed.
So it's active recycling. It's like ripping the cardboard boxes apart so they can actually fit in the bin.
That's a great way to put it. It extracts proteins from cellular structures to hand them off to the incinerator, which is the ubiquitin proteasome system, or it bags them up for the heavy-duty dump, which is autophagy.
Okay, so VCP is a manager of the recycling plant. It decides what stays, what goes, and it does the heavy lifting. Now, what happens when this specific machine breaks?
Well, the manager goes on strike, or more accurately, the machine gets a gear stuck. The paper describes this as a failure of autophagic flux.
Flux. That implies flow, so the flow stops.
The flow stops, and the trash doesn't just sit there. It backs up the entire system. You get these things called immature autophagosomes. Think of them as trash bags that have been filled up, tied off, but the truck never came to pick them up.
And they're just sitting in the hallway.
Sitting in the hallway, leaking toxic goop. And this is where the path splits between ALS and CMT. This is the really cool part. The paper details how different mutations in the same VCP gene can lead to two different disasters.
Walk me through that. How does the same broken machine cause two different diseases?
So in ALS specifically, a type called ALS6FTD, there's a specific VCP mutation, like C.464GA, that causes a protein called TDP43 to clump up.
Right.
Now, TDP43 is the villain in almost all ALS cases. When VCP fails to police it, TDP43 aggregates in the motor neurons of the brain and spine. It's incredibly toxic. The cell just dies, and it dies rapidly.
And when the motor neurons in the brain die, you get the rapid paralysis of ALS.
Correct. But then you look at CMT. The paper discusses a different mutation in VCPC.553GA found in a family with CMT type 2Y. In this case, you don't get that massive TDP43 explosion in the brain. Instead, those trash bags, the autophagosomes, they accumulate. But where do they accumulate?
Then in the brain.
In the long axons of the peripheral nerves, the nerves that are running all the way down to your feet.
So in one case, the central headquarters burns down because of a toxic spill. That's ALS.
Yep.
And in the other, the delivery trucks just get stuck in traffic miles away from the city. That's CMT.
That is a perfect analogy, and that traffic jam brings us to the second major concept this paper explores, long axon vulnerability.
I wanted to dig into this because we hear motor neuron and we think of this microscopic cell, but these things are weirdly huge, aren't they?
They are giants. I mean, we don't think about it, but the nerve that controls your big toe, the cell body is in your lower back, inside your spine.
Yeah.
But the axon, the wire connecting it to the toe, is a single continuous structure.
How long are we talking?
In a tall person, that axon can be over a meter long.
A single cell that is a meter long.
Yes. And to put that in perspective, if the cell body were the size of a tennis ball and it was sitting in Times Square, the axon would stretch all the way to Philadelphia.
Wow. That is a logistical nightmare.
It's impossible logistics. You have to transport energy like mitochondria and take out the trash, these proteins, all the way from New York to Philadelphia through a tiny tube. And you have to do it constantly for decades.
So if VCP, our recycling manager, is only working at, say, 80% capacity because of a mutation,
the trash piles up at the furthest point first. Philadelphia gets messy before New York does. And that's why you see the symptoms in the feet with CMT first. It's a dying back phenomenon. The supply chain fails at the periphery.
But if the mutation is more severe or it affects a different part of the protein,
then the tennis ball in Times Square explodes, and that's ALS.
That makes so much sense. Long axon vulnerability is basically just the reality that supply chains break at their weakest, most distant links.
Precisely. And VCP isn't the only one struggling with these logistics. The paper identifies 12 other genes. If VCP is the sanitation department, well, we have to talk about the other city services.
Okay, let's talk about the supporting cast. Who else is messing up the city?
Well, if VCP is sanitation, we have to talk about SOD1.
SOD1, that's the OG. That was the first gene ever linked to ALS back in, 1993? I remember reading about that.
It was. For 30 years, SOD1 was synonymous with ALS. It's an enzyme that handles oxidative stress. So basically, it prevents the cell from rusting from the inside out.
It neutralizes free radicals.
Right. But the paper throws a massive curveball here. They cite this case report of a 50-year-old woman. She comes to the clinic. She's got foot drop, muscle atrophy in her legs, very slow progression, classic CMT.
So the doctors run the CMT panel.
They do. Negative, negative, negative. They scratch their heads. Finally, they sequence her whole genome. And what do they find?
Don't tell me she had an SOD1 mutation.
She had the SOD1 C.140AG mutation. And so did her daughter. This gene, which we all thought was the grim reaper of ALS, was causing a manageable chronic neuropathy in this family.
That completely blurs the line. That means the ALS gene isn't strictly an ALS gene.
Exactly. It suggests that oxidative stress, the rusting, can manifest as either disease. If the rust is bad enough, the engine seizes immediately, that's ALS. If it's a slow corrosion, the car just runs poorly for years, and that's CMT.
So I've got sanitation with VCP and rust prevention with SOD1. What about the actual roads themselves, the infrastructure?
That's where a gene like NEFH comes in. It codes for the neurofilament heavy chain.
Neurofilaments, those are the structural girders, right? They hold the axon open.
Right. They are the skeleton of that New York to Philadelphia tunnel. They keep the tube open so traffic can flow. If you have a mutation in NEFH, the girders can collapse, or they can clump up and block the road.
And again, if the road is blocked near the end of the line, you get CMT symptoms. If the blockage destroys the central hub, you get ALS.
You're seeing the pattern. It's all about the cellular city. Whether it's the trash piling up, the rust taking over, or the roads collapsing, these are shared mechanisms of failure.
But a city needs power. We can't run recycling plants or repair roads without electricity. So we have to talk about the power plants.
Mitochondria.
The powerhouse of the cell. I think this is the only thing everyone remembers from high school biology.
It holds up. And this is the fourth pillar of the overlap. The paper focuses on a gene called MFN2, mitofusin 2.
Mitofusin. Sounds like it fuses mitochondria.
That's exactly what it does. Mitochondria aren't static. They're dynamic. They dance. They fuse together to share resources, and they divide to multiply. They need to do this to stay healthy, especially at the end of that long axon. MFN2 is usually the textbook cause of CMT type 2A.
Okay, so MFN2 usually means CMT.
Usually. But here's the story from the paper that really stuck with me. It challenges everything we think we know about inheritance. There was a family where the mother developed aggressive ALS. She had frontotemporal dementia and she had bulbar onset, meaning she couldn't swallow or speak.
That's the most severe form.
It is. She passed away quickly. Then they looked at her son. He had the exact same mutation in MFN2, the PSP194ALA1.
So he developed ALS too.
No, he developed CMT. He had sensory motor neuropathy, issues with his feet. But his brain was fine, his life expectancy, normal.
Wait, hold on. Same gene, same mutation, same typo in the source code. Mother gets fatal ALS, son gets chronic CMT. How is that even possible?
That is the multi-million-dollar question, and it proves that genetics isn't just a blueprint. It's a recipe that can change depending on the kitchen. The paper suggests this is where modifiers and epigenetics come in.
Epigenetics, that's the idea that our genes have volume knobs, right?
Exactly. You can have the code, but is it being shouted or is it being whispered? The paper highlights methylation, which is adding a chemical tag to DNA, as a major factor. For example, in ALS, we often see hypermethylation of a gene called C9 or F72. It's like someone turned the volume knob way down.
So maybe the mother had a loud background genetic profile that amplified the damage, or the son had protective modifiers that dampened it.
Precisely. It could be other genes acting as buffers. But it's not just genes. The paper explicitly lists environmental factors. Nutrition, physical activity, toxin exposure.
The environment.
Think about it. Maybe the son was an athlete or had a diet rich in antioxidants that helped his mitochondria cope just enough to keep the motor neurons alive, pushing the pathology out to the peripheral nerves instead.
That is incredibly hopeful, actually. It implies that even with a bad gene, the outcome isn't 100% written in stone. There's wiggle room.
There is wiggle room, and understanding that wiggle room, those modifiers, is how we find new treatment.
Which brings us to the so what of this deep dive. We've established that these diseases are molecular siblings. Why does this matter for the patient sitting in the doctor's office today?
It matters for two huge reasons: diagnosis and treatment. First, let's talk about the diagnostic odyssey.
That's the limbo patients get stuck in when doctors can't figure out what's wrong with them.
Right. Early stage ALS can look a lot like CMT. Muscle wasting, weakness in the limbs. If a doctor sees those symptoms, tests for the standard CMT genes, and finds nothing, they might just shrug. They might say, well, we don't know.
Meanwhile, the patient is terrified.
Exactly. But if that doctor understands this overlap, they might say, you know what, let's check the ALS panel, let's check VCP, let's check SOD1. Finding the genetic cause ends the uncertainty. It helps with family planning. It explains why.
And finding the answer is the first step to fixing it. So, does knowing that VCP is the culprit change the pills you take?
We are moving toward that. This is the shift to what we call mechanism-based medicine. We are moving away from treating ALS or CMT as labels and moving toward treating the failure.
So if the failure is trash buildup, we treat the trash buildup.
Exactly. The paper gets really excited about a class of drugs called HDAC6 inhibitors.
HDC6, what does that do?
So HDAC6 is an enzyme that removes markers from the microtubules, the roads. When you inhibit it, you essentially smooth the pavement, it improves transport, and it helps clear those protein aggregates.
And since both ALS and CMT have traffic jams.
The same drug could work for both. That's the breakthrough. Instead of developing a drug for a tiny sliver of ALS patients, you develop a transport enhancer or a proteostasis booster that helps ALS patients, CMT patients, and maybe even Parkinson's patients.
That makes the market bigger, which means more research money can flow in.
Exactly. The paper also mentions the role of AI. They used AlphaFold 3 in this research.
That's the Google AI that predicts protein shapes.
Yes. They used it to model exactly how those VCP mutations twist the protein. And because they can see the twist in 3D, they can design small molecules that fit into that twist, like a key in a lock.
And potentially fix the machine.
Or at least stop it from jamming. And of course, there's CRISPR. If we know the exact letter change that G to A switch CRISPR offers the dream of going in and editing the typo back to normal.
It's a brave new world. We started with Charcot in the 1800s, looking at patients and putting them in different boxes based on how they walked or how long they lived.
And now we're looking at the molecular machinery and realizing those boxes are, well, they're artificial.
It really challenges how we name things. It feels like we are on the verge of a major rebrand in neurology.
I think so. I'll leave you with this thought. In 20 years, I don't think we'll be diagnosing people with ALS or CMT. Those are descriptions of symptoms. I think we'll be telling a patient, you have a VCP-mediated proteostasis disorder, or maybe you have a mitochondrial transport failure.
And because we name it by the mechanism, the name itself tells the doctor which drug to pull off the shelf.
Exactly. You treat the cause, not the consequence.
That is a powerful reframing. From observing the walk to fixing the engine.
It really is.
Well, that's all for this deep dive. We hope this connected some dots for you, connecting the recycling plant of VCP to the long highways of your nerves. Huge thanks to the research of Einash and Kursula for lighting the way.
And thanks for listening.
Stay curious and keep asking questions about what's written in your DNA. We'll catch you on the next one.